Plants exhibit remarkable growth and development throughout their lives. From a single seed they can unfurl into towering trees or spread into sprawling vines, their forms dictated by an intricate interplay of internal signals and environmental cues. Among these internal signals, plant hormones play a pivotal role, acting as chemical messengers that regulate various aspects of plant growth and development. Apical dominance refers to the tendency of the main, central stem of a plant to grow more vigorously than its lateral branches. This dominance establishes a hierarchy within the plant, with the apical bud, located at the tip of the main stem, exerting control over the growth of lateral buds situated along the stem's sides. The answer lies in the fascinating world of plant hormones, particularly a group known as auxins. Plant growth and development are precisely orchestrated events governed by a complex network of internal signals. Plant hormones are crucial regulators, orchestrating growth and development. Produced in minute quantities, they affect seed germination, root elongation, flowering, and fruit ripening. Major groups include auxins, gibberellins, cytokinins, abscisic acid, and ethylene. Each plays a distinct role, often interacting to fine-tune growth. Among the various plant hormones, auxins stand out as key players in apical dominance. These chemical messengers, primarily indole-3-acetic acid, IAA, are synthesized in the apical bud. Auxins travel downward, influencing growth and development. They affect cell elongation, a fundamental process in plant growth. By increasing cell wall plasticity, auxins allow cells to elongate. In apical dominance, auxins suppress the growth of lateral buds. The story of apical dominance begins at the very tip of the plant, in the apical bud, where auxins are synthesized. This localized production of auxins is crucial for establishing the concentration gradient that underpins apical dominance. Within the apical bud, specialized cells work tirelessly to produce auxins, primarily indole-3-acetic acid, IAA, the most common and potent naturally occurring auxin. Once synthesized, auxins embark on a remarkable journey through the plant, moving primarily in a unidirectional manner from the apical bud downwards. This polar transport of auxins is facilitated by specialized transporter proteins embedded within the cell membranes of plant cells. These transporter proteins act like gatekeepers, controlling the movement of auxins between cells and ensuring their downward flow. As auxins move downwards from the apical bud, they gradually decrease in concentration, creating a gradient with the highest concentration near the tip, and progressively lower concentrations towards the base of the plant. As auxins flow downwards from the apical bud, they exert their influence on lateral buds, dictating their growth and ultimately shaping the plant's architecture. The key to understanding this control lies in auxins' inhibitory effect on lateral bud growth. High concentrations of auxins, such as those found near the apical bud, suppress the growth of lateral buds, preventing them from developing into branches. The exact mechanism by which auxins inhibit lateral bud growth is complex and not fully understood. However, it is believed to involve the interplay of auxins with other plant hormones, particularly cytokinins, which promote cell division and lateral bud development. Auxins are thought to interfere with the synthesis or transport of cytokinins, thereby suppressing their growth-promoting effects on lateral buds. This inhibitory effect of auxins on lateral bud growth is most pronounced near the apical bud, where auxin concentrations are highest. As the distance from the apical bud increases, auxin concentrations decrease, gradually releasing lateral buds from their inhibitory grip. The auxin concentration gradient established by polar transport plays a crucial role in determining the length and vigor of lateral branches. As auxins move downwards from the apical bud, their concentration gradually decreases, creating a gradient that influences the degree of lateral bud suppression. Near the apical bud, where auxin concentrations are highest, lateral buds experience the strongest inhibition, resulting in shorter and less vigorous branches. As the distance from the apical bud increases, auxin concentrations decrease, gradually releasing lateral buds from their inhibitory grip. This release allows lateral buds further down the stem to grow more vigorously, producing longer and more developed branches. The result is a tapered growth pattern with shorter branches near the top of the plant and progressively longer branches towards the base. This gradient of control, mediated by the auxin concentration gradient, is essential for shaping the overall architecture of the plant. Apical dominance, driven by the interplay of auxins and other plant hormones, plays a pivotal role in shaping the diverse forms and architectures observed in the plant kingdom. From towering trees to sprawling vines, the degree of apical dominance influences the plant's overall shape, branching pattern, and ultimately its success in competing for resources such as sunlight and nutrients. In trees, strong apical dominance is crucial for establishing a central trunk that can support the weight of the crown and reach heights necessary for optimal light capture. The tall, straight trunks of many tree species are a testament to the power of apical dominance in shaping plant form. In contrast, shrubs and bushy plants often exhibit weaker apical dominance, allowing for the development of multiple lateral branches and a more spreading growth habit. Vines utilize apical dominance to their advantage by suppressing lateral branch growth until they encounter a suitable support structure. While apical dominance plays a crucial role in shaping plant architecture, it is not an absolute dictator. Plants exhibit a remarkable ability to fine-tune the degree of apical dominance in response to internal and external cues. This dynamic balance between dominance and branching allows plants to optimize their growth and development in response to changing environmental conditions. One way plants regulate apical dominance is through the interplay of auxins with other plant hormones, particularly cytokinins. As mentioned earlier, cytokinins promote cell division and lateral bud development, acting as antagonists to auxins' inhibitory effects. The balance between auxins and cytokinins plays a crucial role in determining the degree of apical dominance and the extent of lateral branching. Environmental factors can also influence apical dominance. For instance, shading from neighboring plants can reduce auxin production in the apical bud, weakening its dominance and promoting lateral branch growth. The interplay of hormones like auxins and cytokinins provides a fascinating glimpse into the internal mechanisms governing plant growth. External factors, the environmental cues that surround a plant, play an equally crucial role in shaping its final architecture. Light, wind, temperature, and nutrient availability all contribute to the symphony of signals a plant must interpret. Light, being the primary source of energy for photosynthesis, exerts a particularly strong influence on plant form. Plants have evolved sophisticated mechanisms to detect and respond to variations in light. Understanding the principles of apical dominance and its manipulation has profound implications in horticulture and agriculture. Gardeners and farmers have long exploited these principles to control plant growth, shape desirable forms, and enhance crop yields. Pruning, a common horticultural practice, is a prime example of how humans manipulate apical dominance to achieve desired outcomes. By removing the apical bud, pruning effectively disrupts the flow of auxins from the tip, releasing lateral buds from their inhibitory grip. This release stimulates the growth of lateral branches, resulting in a bushier, more compact plant. This technique is commonly used in hedges, topiaries, and fruit trees to promote lateral branching and increase flower or fruit production. Similarly, pinching back, the practice of removing the tip of a young shoot, also disrupts apical dominance and encourages bushier growth. In agriculture, understanding apical dominance is crucial for maximizing crop yields. Apical dominance, orchestrated by the intricate interplay of plant hormones, particularly auxins, stands as a testament to the remarkable ability of plants to regulate their growth and development in response to internal and external cues. This phenomenon, characterized by the dominance of the apical bud over lateral bud growth, plays a fundamental role in shaping the diverse forms and architectures observed in the plant kingdom. From the towering trunks of ancient trees to the spreading canopies of forest giants, apical dominance shapes the plant's ability to compete for resources, withstand environmental challenges, and ultimately thrive in its ecological niche. Understanding the mechanisms underlying apical dominance provides valuable insights into the fascinating world of plant growth and development, highlighting the intricate connections between genetics, hormones, and the environment. As we continue to unravel the complexities of plant signaling and response mechanisms, we gain a deeper appreciation for the remarkable adaptability and resilience of these organisms that form the foundation of our ecosystems and sustain life on Earth.